All right, I'm going to start. So today's lecture is about phylogenetics. Hopefully it won't run as long as last time. And I'm also postponing homework associated with this particular lecture because homework seems to be piling up on you guys. So we'll do this one a little later on when there's a little more room in the schedule. Last homework from last time, the motifs, I think you should be able to get it done. If there's huge problems, write to us and let us know. Let me know that there's a problem with the homework. But otherwise, hopefully, it can be done by Tuesday next week. All right. So the main points that I plan to cover today, talk a little bit about molecular evolution, just an overview of the biology that underlies and motivates us to study phylogenetics. Then we're going to talk about calculating distances, which is really a rehash of what we talked about in making multiple alignments. And you'll see a lot of parallelism between the whole process of multiple alignments and the whole process of building phylogenetic trees. We'll talk a little about clustering algorithms, a little about cladistic methods. Phylogenetic theory gets very deep, and I'm not going to try and sort through all the details in just one lecture. I just want to point you at the fact that there is a lot of information and some controversy there. And then we're going to review some software tools that you can use in your everyday work to make trees because they are important tools in molecular genetics. So evolution is what we're talking about when we're talking about phylogenetics, the foundation of all modern biology. From a molecular perspective, sequences change over time. Organisms change over time, but we're looking at their DNA sequences and how those change over time. Also, obviously, we're looking at protein sequences. And time is always a factor in phylogenetics. And in fact, whenever you're looking at a comparison of two sequences, you're doing a blast search or some other form of sequence analysis, you always have to take into consideration the time factor. How have these two sequences changed over time from some common ancestor? That's actually implicit in all the assumptions that are made when you do an alignment or a multiple alignment or any other kind of sequence comparison. So nothing in biology makes sense except in the light of evolution. Theodosius Dostoevsky, really concise, and if you don't have evolution as your foundation, then none of your molecular genetics make any sense. So when we're talking about similarity searches and alignments, multiple alignments, we're always implicitly asking the question, how are these sequences related? And also, we may be asking the question, how are the organisms from which these sequences originate related? So what's the relationship between mouse and human when we're comparing a mouse gene with its orthologous human gene? And sometimes that relationship is tight and solid, and other times the relationship is more confusing. The history of the genes may not be exactly the same as the history of the organisms. So we can make statements about the genes more easily than we can about the organisms because we have the sequences, we can compute their distances. The conclusions that we make about the organisms are inferences. Oops. So wow, that's funny. So I just wanted to show an example. So this is what I mean when I'm talking about a tree. I'm going to define it more carefully later. But the idea is that things that are connected by short branches are more closely related to each other, and things that are connected by longer branches are more distant. So the ECP human and Pohnpei, which I guess is a monkey of some sort, is more closely related, and then ECP2 mouse and ECP1 mouse are more closely related, and then there's this rat gene that's not in a close relationship with either of the other two. So that's the kind of inference you could make from reading a tree. Wow. So the purpose of the tree is to illustrate how a group of objects are related to each other. Just a very, very simple conceptual level. You know, we're going to talk about math, but always it's to prove a point. The A is closer to B and C is further away from A and B. That's the reasoning, and you iterate that reasoning over whatever set of sequences or that you have to work with. So phylogenetics is also a mapping of this more general concept of taxonomy into computation. Taxonomy is a way of organizing things. You have things that fall into classes, and then classes that come together into groups, and groups that come together into supergroups or families, etc. Taxonomy goes back to Linnaeus in the early 1700s, and fundamentally it's a very human idea. You can give somebody a pile of nuts and bolts and screws and say, organize them, and that person will come up with a taxonomy. It may not be exactly the same as another person with a similar pile, but putting things into classes by their similarities is just a way of thinking and organizing the world. And so naturally we organize living things into some sort of a taxonomy. And Linnaeus actually had this concept of the tree of life, that everything is related. You know, all living things have some common ancestor, which was an important fundamental insight, especially in the 1700s. And you know, it sort of rules out the concept of separate creation, that different living things are created from completely different origins. And you know, from a molecular standpoint, we have ample evidence for shared ancestry for all living things. So phylogenetics is a little bit more of a mathematical approach to the concept of taxonomy. And it basically states that groups of similar, that evolution should be the fundamental driver of your taxonomy, that groups should share common ancestors. So it's not just that you group things conveniently into four-legged and six-legged and no legs, but rather that all the members of a given group should share a common ancestor, and the members of another group should share a different common ancestor, and they shouldn't be mixed. Phylogenetic systematics, also known as cladistics, is a method of taxonomic classification based on evolutionary history. And this was developed by the entomologist Willy Henning in the 1950s. And it represented, you know, a more rigorous mathematical approach to taxonomy. So in a cladistic method, you have features. And those features could be anything that you want, any sort of phenotype that you can observe about your organism. And you consider various possible pathways of evolution for those features and choose from them the best tree, the one that brings together the organisms by sharing of conserved features. Of course, that requires a judgment call as to which features are conserved from an evolutionary standpoint and which ones are what they call derived, meaning more recently acquired. So like the concept of, say, eyelessness in a cavefish, that's a derived character, and other eyeless organisms are probably not closely related by ancestry. Rather, you know, the concept of having a backbone is probably a more evolutionarily conserved feature, and all things that have a backbone probably all derive from a single common ancestor. So the work of taxonomists was quite challenging because they had to make a lot of judgment calls for a lot of organisms, all of them essentially, as to which features were more fundamental and which features were more recently acquired and therefore not useful for making deeper taxonomic divisions. A cladistic tree has branches that are proportional to the evolutionary history, and a phylogram is a tree that has branches that have specific lengths related to evolutionary distances. These things make a lot more sense when we start computing with sequences, because lengths then become proportional to sequence changes. So just an example, a cladogram in the upper part of the graph here shows the relationships. These bacteria are related to each other, these eukaryotes are related to each other, but the relative distances are not meaningful. The phylogram down here is meant to be to have important distances. So the length from one organism to the next following the tree represents the total number of sequence changes going from one organism to the next for whatever piece of sequence was used to calculate this tree, or whatever set of characters, I should say, in a more general sense. So phylogenetics often makes use of numerical data, but initially that numerical data was sort of a tabulation of characters where all the characters were considered equally. And so you were looking at, whether these two organisms shared, 20 out of 100 characters. And those characters could be anything: resistance to an antibiotic, having a backbone or not having a backbone, having a cell wall, whatever the taxonomist chose to include in that list of characters. And then you could do math on that matrix of ones and zeros. Using DNA sequences as a phylogenetic character greatly improved the science for a lot of reasons, one of which is that DNA is quite objective, right? The sequence of a particular gene from a particular organism can be determined to a reasonable level of factual accuracy. You could send the same sequence to multiple labs and confirm that it's correct. Whereas taxonomic characters often have a little bit of judgment call. Does the fruit fly have long or short spines on its abdomen, that sort of thing. Also, there's a very large number of possible characters when you start working with DNA sequences. Potentially any sequence in the whole genome of an organism, any base, can be a character. Frequently, you focus on a particular gene of interest or even just a few taxonomically relevant characters. But if you need more are available. Okay, so yeah, I said this all. So characters are unambiguous, large number of characters, and information about the extent and nature of divergence is available. Some types of changes in DNA are unlikely to occur by chance in separate lineages. So if you have a particular deletion or fusion of two different pieces of DNA together, that's a character that's likely to be passed on to progeny and very unlikely to occur in a separate instance in an unrelated group of organisms. So there's both distance type measurements, the count of bases that differ between any two sequences, and then there are these sort of larger, more unique, distinctive characters that can be inferred from DNA. Okay, so I'm going to try and define characters a little more precisely. So, as might be intuitively obvious, you can only work with DNA characters in the context of an aligned group of homologous sequences. You need to have the same sequence from every organism that you're studying, and you need to make a multiple alignment because you're going to count differences, and differences are only relevant when they are differences in truly related sequences. Usually we like to use the word orthologs. I'm going to define that a little more carefully in a few slides. But here we have a little segment of a multiple alignment, and I promise that these are orthologous, homologous sequences from different organisms. The way I've written out the matrix, wherever the sequence is identical in B, C, and D, I've replaced the letter with a dot. So the dots mean no change here. And as you can see, some of the columns, some of the positions have useful information that allows us to make taxonomic decisions. I mean, because the goal here is to draw a phylogram, right? To figure out which pair of sequences is most close, and then which one is next, and which one is next, and draw one of those branching tree diagrams. It's not helpful if you're in the, whatever it is, the 9th column where every base is different in this alignment, because then there's no sharing of the features that bring one pair of sequences closer than another pair. Intuitively, if we had more than 4 sequences in our alignment and we were looking at DNA, we run out of characters and they are going to repeat. And so that wouldn't be the case if you had 100 sequences lined up. There would be some letters that are shared and some that are different. Yeah?
I think in B, there's an A, third letter.
I'm not getting what you're asking. Oh, yeah, right. Typo. Yeah. So this should be a T in order to make the point that I was trying to make. Fair enough. Yeah.
So, are you saying that it's very important to have this in the?
Well, no, what I'm saying actually is that where there's no differences, there's no information. I mean, highly diverse sites can have information. So let's minimize the talk about not having any that are the same. It's more important the ones where all the letters are the same, it's A all the way down. That is what we would call an uninformative site. And you can only use the informative sites where there are differences in the alignment. And that will help you say, well, in this position, right, A is farther and B, C, and D are all closer together. In this position, B is farther and A, C, and D are closer. And you could start counting these things on your fingers, but obviously a computational solution presents itself. I mean, there's an algorithm here that can count the number of letters shared between each pair of sequences and create a distance matrix. Which pair of sequences shares the most letters, which pair of sequences shares the least letters, etc. And that's our goal, at least in the first round. Yeah? We'll talk about that in a little bit. At the DNA level, it doesn't matter. It's still a taxonomically relevant change. You can, when you're working with protein alignments, you can do taxonomy on more distantly related sequences where it would be difficult to make a good alignment with the DNA sequences, or the distances might be very noisy. But very frequently we want the additional resolution that comes at the DNA level. You can do this taxonomy with proteins or DNA, but it's just frequently difficult to get very good DNA alignments. So in those cases, you work with the protein level. If you're working at sort of the species level and lower, then you're almost always trying to get the maximum amount of information to differentiate your samples, in which case you would work at the DNA level. There's also a number of situations where you first align the proteins and then you replace the amino acids with the DNA sequences that code for it. So that's a sort of a way around that problem. Yeah.
So is there a minimum length that you want to look at?
Well, it's like more phylogenetic information is good. So very frequently you'll have unresolved differences where this and this and this are all the same distance apart. So then you need more information to resolve them better. Okay, so the sequences obviously reflect the relationships of the organisms, or at least they reflect the relationships of the sequences. They have distances from each other. They also have perhaps patterns of large-scale sequence changes. And you get an intuitive feel for that if you work with sequences for a while. You do a lot of blast searches. You'll see that organisms that are similar to each other have higher e-values, and more distantly related organisms have lower e-values. There's a lower percent similarity or lower percent identity between the sequences of more distantly related things. So, so we're working in a space where there's a certain intuitive sense to it, as well as carefully computed phylogenetic values. It's important to think about this in terms of, given a set of sequences, it should be possible to reconstruct the evolutionary relationships of those sequences, and therefore of the organisms that those sequences come from. So we know what our goal is, and it seems intuitively to be possible. So we should find a computational method that achieves that goal. And so here's what I'm talking about. We've got a small set of sequences. They share a certain number of bases. They differ in a certain number of positions. And from that set of sequences, we could build such a tree, where the shortest branches represent the sequences that are most similar to each other, and therefore the organisms that are most evolutionarily similar to each other. And as you go backwards through the tree, the distance, the amount of time that's passed since the last common ancestor increases, right? So that's our goal, is to build a tree from the sequences that reflect the evolutionary relationships of the organisms. I'm going to interject here for a moment that it matters what sequences you choose. I've already said that you can only build a tree from homologous sequences, right? You have to have the same sequence. It codes for the same thing, be it a protein or something else. A 16S ribosomal RNA would be an example of a non-protein coding sequence that makes a good phylogenetic tree. What sequence you choose will influence the result of your analysis. That's obvious. And so how do you choose a good sequence that will give you a reasonably accurate phylogeny? Different sequences in the genome accumulate changes at different rates. Highly conserved sequences that have important biological functions that are tightly constrained by shape, therefore are unlikely to vary much over time, will allow you to make phylogenetic trees for more distantly related organisms. Sequences that change very rapidly will allow you to resolve phylogenetic trees for very closely related organisms, such as strains or even different individuals within a population. So depending on the goal of your phylogenetic analysis, that will guide you to what sort of sequences you want to use. So just in general, protein coding DNAs are constrained by natural selection, so they are better for more distant relationships. Highly variable sequences like immunoglobulins, ribosomal RNAs, certain kinds of simple repeats, simple sequence repeats, change very rapidly, sometimes at the level of individuals, so that even people in the same family will have sequence differences. So those would be good for working with closely related organisms. Different regions within the same gene can evolve at different rates. We've been looking at that in terms of the multiple alignment. There's conserved regions and variable regions in a set of aligned proteins. So that can impact your study. I'll give you a heads up that many of the more recent phylogenetic taxonomy studies where people are trying to reorganize a group of organisms don't rely on a single sequence. They tend to take a set of proteins that they think are good and characteristic for the group of organisms that they're working with that have a level of variation that's suitable for the size of the taxonomy they're trying to build. So if you're reorganizing some subclade, then you need relatively rapidly evolving sequences. If you're trying to reorganize all the archaebacteria, then you need very slowly evolving sequences. But they tend to take a group of such sequences, concatenate them together into a supersequence, and then do the phylogeny on that, hoping that basically any anomalies that affect the evolution of one particular sequence in one or a small group of organisms inside of the tree get averaged out by other genes that have had a different level of selection and sequence change. So a concatenation of 3 or 5 or 10 sequences is probably going to give you a more accurate average tree that more accurately reflects the evolutionary history of the organisms rather than just the evolutionary history of that one gene. Okay, now I define orthologs and paralogs. So when comparing gene sequences, it's important to distinguish between identical versus merely similar genes in different genomes. Orthologs are homologous genes in different species with analogous functions and a single common ancestor. So it's basically same gene, same role, different species. So, any example I give is going to be controversial, but let's just say actin, right? So there's a mouse actin and a human actin and a frog actin, and they should all be orthologs. Paralogs are similar genes that are the result of internal gene duplication within a single genome. This happens a lot over evolutionary history. In fact, if you look at PFAM, which is a collection of protein families, essentially by definition, all the members of a given family that are in one species are the results of duplications. The globin genes are a good example. I'm going to show you a slide in a second. So if you have a phylogeny, if you've built a tree, and some of the members of that tree are orthologs, and others are paralogs, duplicate genes that have a separate evolutionary history, then your phylogeny is going to be messed up. It's going to be wrong. As a corollary to that, sometimes a phylogenetic analysis is the best way to identify paralogs and sort them out. So if you have that objective in mind when you build your tree, you'll be all right. If you're trying to classify organisms and you've got some myoglobins and some hemoglobins, each one representing a different organism, you're going to get a wrong tree. I will show you an example in the globin family, right? It's some early ancestor, eukaryote probably. There was a single ancestral globin gene. And at some time, that globin gene underwent a duplication. And so now you had a hemoglobin ancestral and a myoglobin ancestral. Now that there's two different copies of this gene in the genome, those two copies are free or do in fact undergo sequence changes independently. And the change of sequence of one gene does not affect the change of sequence of the other. Over time, these two different genes and the proteins they encode can come to be sufficiently different that they now have different functions. in parallel, you may also have changes in regulatory sequences which cause these two different copies to be expressed differently. But unless you're using regulatory sequences in your alignment, that's not really relevant to our discussion here. But then those two genes are now in some ancestral organism, and a speciation event occurs. And, you know, primates separate from rodents, and now we have a human with a myoglobin and a hemoglobin, and we have a mouse with a myoglobin and a hemoglobin. If you built a tree that from the mouse grabbed the myoglobin and from the human grabbed the hemoglobin, thinking, well, these sequences are similar, they have a good blast value, I will build a tree from them. That tree would not represent the evolutionary history of human versus mouse. It would take you all the way back to this ancestral gene duplication time when hemoglobin and myoglobin had one ancestral gene. So you'd be jumping back in time, you know, 200 million years rather than 50 million or whatever. You'd be going back 1 billion years rather than 200 million, something like that. So you get a very wrong tree. And then if you had sort of a random collection of hemoglobins and myoglobins from a bunch of different animals and you built a tree, the tree would be all screwed up. So you need orthologs. You need to be sure that you have the same gene that is currently being used for the same function in all of the organisms, or in all of the sequence entries in your data list that you're going to use to build a tree. Okay, so I'm going to give a disclaimer before I get into the software. Mostly the point is that phylogenetics is a controversial area, and it has been for a really long time, and that's not likely to change at any point in the future. Different people have different ways of doing this that will contradict each other, and they just continue to argue it out. Neither the theory nor the practical application of any algorithm is universally accepted, meaning you have a data set, you grab some good, well-cited software, you use a method that has a citation on it, you send that in, and some reviewer writes back and scathingly tears you apart because you should have used this other method that was written by his friend and postdoctoral associate. You know, people have opinions, and any tree that you assert given a method is going to have a critic. So that's bad. Also, you have a data set, you open up a software package, you choose a method from a menu, it produces a result. Doesn't mean it was done correctly. Just because the software is willing to open your data and you have a menu option that's available to you does not mean that's theoretically or conceptually the best way to analyze that data. So you need to be a little thoughtful and careful about how you do this work because people can criticize it. And you will get results. That's the thing about working with phylogenetics. Given a set of sequences and just a moderate amount of bioinformatics skill, you can produce a tree that looks authoritative. But your methods may not be the best possible way of getting an answer that you hope to get. It can be messy like this. Also, things that we think we know in terms of taxonomic relationships of organisms are constantly being revised. I mean, seals and sea lions are no longer in the same group. As of 1983, people decided they needed to split the taxonomy of this whole area of carnivores and reorganize it fundamentally and create a brand new group for the sea lions. So, the things that you think you know are not necessarily true, and people can constantly, either using new data or new computational approaches, revise taxonomies and phylogenies. So, again, it's a controversial area. I've said this before, but one more time, when you do a phylogenetic analysis, sort of by definition, you're working with a set of sequences. Those sequences represent genes. We hope they represent the species from which we have isolated the DNA, or from which somebody says they have isolated the DNA, but it's not necessarily true. The gene itself, the piece of DNA, may have a different evolutionary history as compared to the organism it's in. There may be some horizontal gene transfer, which can be some crazy virus-mediated thing, or it can be a simple hybridization where there was a crossbreeding of different subspecies, and that formed a hybrid which integressed back into the main population of species B, and so this gene moved across. I mean, when they talk about Neanderthal genes in the human genome, that's what they're talking about, is an introgression. Also, genes experience selection. A particular gene can, in a particular lineage, subgroup within a larger family, can evolve more rapidly under pressure. This particular group of organisms needs to develop some resistance to some chemical in its environment, and it changes whatever protein was the target for that chemical. There's very high selection pressure, and it changes rapidly. And in another group, that same protein does not change so rapidly. So now the trees that you calculate using that particular protein will be quite different than trees you might calculate using a different protein that did not undergo that very rapid selection and sequence change. Okay, there are two branches of phylogenetic methods. There are the cladistic methods and the phenetic methods. The cladistic methods are more theoretically complex, and very frequently they can give you better trees. The phenetic methods are more theoretically simple and easier to explain, and I will spend more time talking about them, and more of the molecular biology literature relies on those methods. More of the deeper evolutionary biology literature relies on cladistic methods. So phenetic methods construct phenograms, obviously, and they consider the current data only. They measure differences. They work generally with a distance matrix. So every sequence is a certain number of base changes or amino acid changes from every other sequence in your data set, and you can build a tree from that count of differences. It's a straightforward method. It's computationally efficient, and it'll get done on a laptop in a few minutes. A cladistic method relies on a rich set of assumptions of ancestral relationships. What base change happened first and which one came later? And those assumptions can be quite deep, and they can be built using a variety of different methods. And so I'll talk about that a little bit, but not to the extent that it deserves. So you're not just relying on summarizing all the differences between your sequences as a single number when you're doing a cladistic method. In fact, every position, every change has its own tree, and then you're summarizing all that data together in some algorithmic way. All right, Darwin was a cladist. Quote, "The natural system based on descent with modification, the characters that naturalists consider as showing true affinity are those which have been inherited from a common parent. And insofar as all true classification is genealogical, that community of descent is the common bond that naturalists have been seeking." Summary, ancestry is the way to organize a taxonomy. Not by number of shared features, but by the actual history of what came from what. Who was the common ancestor? Obviously, that's more difficult to define than just counting up the features that are shared. We don't know the ancestors. We don't know the sequences that the ancestors had. We have to infer them somehow from the data that's available to us, with very, very, very few exceptions where, miraculously someone can sequence some ancestral DNA. So we're going to talk about the phenetic methods. Computer algorithms based on phenetic methods rely on distances to build trees. So the phenetic method counts each base difference equally. That's the fundamental idea. Every base difference is just as good as every other base difference. Phenetic approaches lead to faster algorithms, and they often have nicer statistical properties for molecular data. The phenetic approach is popular with molecular biologists because it relies heavily on sequence data and is relatively easily calculated, and it requires few assumptions. The failing point of many of the cladistic methods is that there are many assumptions which cannot be proven to be true for any given data set. Assumptions like no hybridization and all the things that go with that kind of thinking. That every lineage is distinct from every other lineage and there was no mixing at any time in evolutionary past. We can't prove an assumption like that.
So phenetic methods can only go back so far compared to cladistic methods. So when I think of cladistic method, I'm thinking of like a gigantic tree from the dinosaurs up till now.
You can use a phenetic method on any data set, but clearly your accuracy and reproducibility declines as time gets greater. And that's definitely the strength of the cladistic approach, is that they're attempting to tease out what happened first and what happened later. So not every base change is equivalent.
So are there molecular approaches for cladistic?
Yes, there are. Yes, there's a lot of them. And that's where things get more controversial. So we're going to talk about distance measures first because it has such a nice parallel with what we did in multiple alignment with talking about clustyle. So you can measure the genetic distance between two species, two populations, two individuals, or more generally between two sequences. You calculate the pairwise distance from the alignment. And then you calculate a distance metric. So just in general, a distance method gives a single measurement, number of bases changed, between any two sequences. And that is an assumption that they are the number of changes that happened since those two sequences diverged from their common ancestor. That's the assumption. It's not a super strong assumption, but that's the one that we make. So you have your sequences, you compare A to B. You can think of this as just like a Smith-Waterman or a BLAST, where there's a single number that represents the number of differences in the aligned sequences between A and B, A and C, A and D, etc. So eventually you get the distance matrix, right? Between 1 and 2 is 48 differences, between 1 and 3 is 45, between 3 and 4, etc. Some pair of sequences in this matrix will have the shortest distance, the two sequences that are most similar to each other. And those two get the shortest branch. Then comes the next one. Sometimes there's a sub-branch here. These two are shorter, and then both of them are further from these three. And that's your cluster building algorithm. And it's very straightforward. Once you have the distance matrix, you can build your cluster directly. Right, it's simple to compute the difference. It's the sum of all the base differences. Obviously, you need a good alignment in order for this to work. If there's big gaps, you have to have a gap counting algorithm, just like BLAST or Smith-Waterman has to. Generally, all base changes are considered equal. You could have a different score for a transition versus a transversion, large side chain of nucleotides versus small ones. It's not often done in this framework. Usually all base changes are considered to be equal. They are events. something happened. This sequence changed. Insertions and deletions have a larger weight than replacements. Also, this is sort of important. There's this concept of multiple substitutions. Over a larger amount of evolutionary time, you have a mutation at a given position, and then later on, another mutation happens at that same position. Using the simple algorithm, you just see one change. You don't see the fact that that position changed twice. It's actually relatively straightforward to calculate this because the pair of sequences that has the most observed changes will also have the most hidden changes. So it simply makes all the long branches longer. It's a proportional adjustment, right? If you have, if two sequences differ by 50 changes, and then another two sequences differ by 25, logically, the one with more changes also has more double substitutions that have been hidden. Oops. So, yeah, I'm going to skip that for right now. This is about maximum likelihood. I'll get to that in a bit. So, if we're working with amino acids instead of DNA, then we can use an amino acid distance matrix, as we've been doing in the past couple of lectures, right? One amino acid change is not necessarily equivalent to another because some are conserved and some are less conserved. So that can give your distance matrix a little bit more of a functional or evolutionary flavor, that these are conservative changes and therefore the evolutionary distance is smaller. You can do that or not, basically. You have the choice of just using a strictly phonetic method or a method that takes into account amino acid distances. So we end up, oh, look at that. I fixed it and it unfixed itself. Cool. All right, so this is the PAM matrix with a few things shifted. We've seen it a bunch before. Negative scores represent amino acids that are less likely to replace each other than you would expect by chance in a set of conserved positions. Positive scores, which are relatively rare in the matrix, are those amino acids that replace each other more likely than by pure chance at conserved positions in an alignment of conserved sequences. So my example is isoleucine, leucine, and valine. Functionally very similar amino acids. They also happen to be only separated by one base codon changes, and they get positive scores because it's actually an indication that two sequences are closely homologous when they have a change like that, because it's a conservative change. It means the protein has retained its function despite receiving a DNA change. Okay, I'm not going to take too long to talk about how clustering algorithms work, but when you're starting with a distance matrix, it's relatively simple. They construct a tree by linking the least distant pair of taxa followed by successively more distant taxa. The most common and actually best clustering method is called neighbor joining. UPGMA is the simplest. It just takes the closest pair, unweighted pair groups, right? So it takes the closest pair with no weight and makes a branch, and then the next closest, and then the next closest, and the next closest. So it's very, very simple. So lots of methods use that. Neighbor joining corrects UPGMA assumption that the same rate of evolution applies to every branch of a tree. So if you have a big tree and there are subfamilies, the sequences could evolve at different rates in those different branches. And it makes a correction for that. So the distance matrix is adjusted for different rates of evolution in each taxa. And just in general, neighbor joining performs well in simulations and benchmarking studies, and it's widely used in many published trees. It's very simple. It's built into the Cladistic method and a bunch of other software. So it's relatively easy to use a distance metric and create a neighbor joining tree. And oh, that's so pretty, right. So just in general, the two closest sequences are formed into one branch, and then of all the remaining sequences, whichever distance is next smallest, those two form a branch, and then whatever distance is next smallest, those two form a sub-branch until you've gone through the whole tree. Okay, I'm going to talk a little bit about cladistic methods now. For character data about the physical traits of organisms, stuff like morphology of organs and deep levels of taxonomy, cladistic method is almost certainly superior. You are losing a lot of information when you take a distance method. You're considering every change to be equivalent, and you're summarizing everything down into just one number as the comparison between each pair of sequences, without thinking about the specific path of evolution that led to those sequence changes and which changes happened in which organisms at what time. As I said, cladistic methods are difficult to implement with molecular data because all of the assumptions are not satisfied. So you're working in a space where you're applying a method to some data, and you know that you can't support all the assumptions that that method requires. So it's your best guess whether the result from that method is valid, and that's why it's so controversial. So cladistic methods are based on the assumption that a set of sequences evolve from a common ancestor by a process of mutation and selection without mixing. In other words, no horizontal gene transfer, no hybridization. The phenetic method doesn't care about that stuff. All it says is that this is the distance we observe. There's 26 base differences between sequence A and B and 37 between A and C. Therefore, A and B form a shorter branch and A and C form a longer branch. The cladistic method makes additional assumptions. The cladistic method works best if you have a specific tree or a known ancestral sequence. Then you can trace the path by which the bases have changed. And you could have a finite number of possible trees to select from. Whereas if you only have the final resulting data set, there's a very, very large number of trees that you can build from even a moderately sized data set. You know, if you have, say, 20 sequences, if you map out the topology of all possible trees, it's really huge. Way more than a power function. Parsimony is the most popular method, or at least it was, for reconstructing ancestral relationships. And at least it's fairly intuitive. It makes decent sense. And I will try and explain some of the guts of it. Advantage of parsimony is that it uses all of the known evolutionary information in building a tree. It doesn't squish down all the information into just one number. It pays attention to every change in every base, and it attempts to guess the history of changes that happened from the ancestor to generate the current data set. So parsimony involves evaluating all the possible trees and giving each tree a score based on the number of changes that are needed to explain the observed data. So this is a minimum evolution approach. So assuming an ancestor, how many changes would have to happen to go from that ancestor to the available data that we have? The tree that requires the fewest changes is, by the definition, the best tree. So, example. Simple example. Some years I get this better than others, so bear with me. 4 sequences: ATCG, TTCG, ATCC, and TCCG. So this is an example of our current data. We're going to imagine a tree that has a known ancestor. So we're like hugely cheating here, because given these 4 sequences, there's a very large number of possible ancestors, right? At least 2 to the 4th possible ancestors. Some of those ancestors are very unlikely because they don't have any of the bases that are present in the current sequence, in the current positions, right? So if I had a G, A, that would be a very unlikely ancestor for these sequences. But postulating a whole bunch of trees, we'll just look at 2 of them, both assuming the ancestor ATCG, which happens to be one of the sequences that we still observe. So there's no changes coming down the left-hand side, right? ATCG goes to ATCG without any evolutionary changes. So there's no cost on that branch of the tree. On this branch of the tree, there's 1 change from ATCG to ATCC. On this branch of the tree, well, we put a T early. So ATCG changes to TTCG first. And then there's a second change here from TTCG to TCCG, right? So there's 2 mutations in this tree. This tree postulates that this C change happened first. ATCG goes to ATCC. Then ATCG changes to TTCG on this branch, and it doesn't change on this branch. And then a 3rd mutation happens from TTCG to TCCG here. That's 3 mutations on the right, 2 mutations on the left. The left-hand tree is more parsimonious than the right-hand tree. I know you don't get it, but the general concept is there. And if it was a much bigger, more complicated data set, still, there's a computer algorithm that could count the total number of mutations required implied by each tree. And even with 4 bases and all the possible ancestors and all the possible intermediate sequences, you could evaluate all the possible trees and find those trees that have a minimum score. Yeah?
Is it basically doing like the explanation that's the easiest is the explanation that's right?
Yeah, it's Occam's razor. The fewest evolutionary events is what must be true. It's not a bad assumption, it's an assumption. Now, if you had additional data, you can build a specific tree or a portion of a specific tree and then let minimal evolution go from there, right? If you knew one of the ancestors in a complicated tree, then you could say this must occur here. And that reduces down the universe of possible trees a great deal. If this gets longer, if you have 100 bases or 1,000 bases, the total number of possible trees and possible ancestors to be evaluated gets out of hand. And so this is a pretty computationally aggressive method. Yeah?
Isn't it possible to get the same score?
Yes.
Finally?
You get many trees, yes.
Like many trees that
Many equivalent trees.
all of these ones?
Yeah, you have many trees. Hopefully some of those many trees have shared branches. And so you can generate a consensus tree for all the places where they agree, where all the trees with the lowest score have same branches. Those are highly supported branches. And I'll talk about bootstraps, which is a similar approach but makes it worse. But helps you narrow down the result. It makes it worse computationally, but it helps you narrow the result at the end. So that's coming like 8 slides down. Yeah?
So let's say you didn't know that ATCG
Right, you have to evaluate every possible tree with every possible ancestor. But some ancestors are very unlikely. As I said, like the AAAA, that's a very unlikely ancestor because any tree that starts there is going to require many mutations to achieve the set of final sequences that we see. So as a starting point, all of the observed sequences are possible ancestors. And you could go from there. And there are what's called branch and bound heuristics for the minimum evolution method, which attempt aggressively to narrow down the space of all possible trees that you need to evaluate by only choosing reasonably likely ancestors. And you can build the ancestors up one level at a time. You don't have to start at the top and work down. You could say, okay, I have ATCG and ATCC. What are the likeliest, most likely possible ancestors for those two sequences? Well, the most likely ancestor is one or the other of them, which would require only one mutation, right? So if you had a longer sequence, you could still make a most likely ancestor or a few most likely ancestors. So it's computationally expensive. There are a number of optimization methods that have been applied to try and make this thing work in realistic amounts of compute time. It doesn't work well if you have thousands of sequences, unless you have hundreds of millions of hours of compute time at your disposal. Some people are doing that, using big superclusters to do this kind of thing. It might make more sense to first do a neighbor joining tree of those same sequences, use that tree as the proposed ancestor, and then try and optimize with the minimum evolution method, rebuilding each subset of the tree, trying to generate, least evolutionary cost subbranches from a known tree. So that works better. And some of these heuristic methods approximate that as well. All right, maximum likelihood is an even more complex and computationally intensive approach to this problem. Maximum likelihood tries to break down the individual evolutionary events even farther. So now, instead of just paying attention to whatever substitutions happen, it tries to use a probabilistic function for those substitutions. How likely is a G to change to an A at this position, given priors? What's the prior? Oh, it might be the GC composition of the organism. It might be the transition versus transversion rate that we've observed in all data, or in this organism, or in some other source of data. So it's an open-ended sort of Bayesian approach where you can bring in whatever data you want and assign frequencies and probabilities to any of the events that happen in the tree. Because you're doing that, it expands the computational world quite a bit. So instead of just evaluating the number of substitutions, now you have to evaluate the likelihood of those substitutions. Even with simple models of evolutionary change, the computational task is enormous, making this the slowest of all phylogenetic methods. Personal observation, I've been working with sets of 70 to 100 cytochrome P450s, and you can do a neighbor joining in about a minute on a Mac. You can do a minimum evolution in about half an hour, and I've never completed a maximum likelihood. I give up after a couple days. Okay, so this idea of parameterizing, adding in values for the different possible changes, is a maximum likelihood approach. So parameters are different substitution rates for each of the four possible, there's more than four, all the possible nucleotide changes from one to the other. I guess there's 12 possibilities. A correction for the proportion of sites which are unable to change. A correction for variable site rates at those sites which can change. And you can estimate these variables, these parameters from the data using fairly complex and sophisticated methods. That's all I'm going to say about that. So maximum likelihood has this interesting property, and parsimony as well, that it predicts ancestral sequences at all the different nodes going backwards up the tree. So that can provide interesting information about the timing of the acquiring of a novel trait. There's a piece of software called PAML, phylogenetic analysis using maximum likelihood, that's popular for this approach. And, well, you can use it. It's free. So some other assumptions that maximum likelihood may use. The frequency for transitions, C to T and A to G, and transversions, C or T to A or G, are different. Assumptions for protein sequence changes come from the PAM matrix and are quite likely to be violated in real data. If you think about it, the PAM matrix is an average for conserved sequences that have known functional domains. Those same amino acid conversion frequencies are not going to apply to every gene, right? Actin genes may have very different evolutionary pressures on valines than whatever average set of genes was used to build the PAM or the BLOSSOM matrix. No group of genes is going to have exactly average amino acid change rates for all of its amino acids. So those assumptions are just, blatantly invalid, but you can use them anyway. All right, I'm going to skip that. So, interesting thought is this notion of a molecular clock. For a given protein, the rate of sequence evolution is constant over time, which means that the number of differences between the gene in organism A and organism B gives you a measure of how long ago they had a common ancestor. Very controversial, very intuitively appealing notion, very controversial when applied to different domains of organisms. Do all organisms evolve at the same rate? Do elephants evolve at the same rate as bacteria, even though their generation times are rather different? Can an elephant acquire as many mutations in 1,000 years as a bacteria does? Yeah. About what? Bottlenecks. So that's a speed up, right? A bottleneck creates more rapid evolution, more rapid sequence changes because you're dealing with a small number of organisms, and also selection pressures can be greater, right? If you die, your sequence is more likely to be eliminated from the pool than if you have a very large population. So yeah, that's the exact kind of thing I was talking about back in neighbor joining when I said it is UPGMA assumes that all the branches of the tree, all the organisms in your taxonomy evolve at the same rate. Neighbor joining says that any subgroup can have a changed rate. So that subgroup might be a group that went through a bottleneck. So yeah, that invalidates the general assumption of the molecular clock in the sense that the group that went through the bottleneck is going to have more changes than some other group. That's the notion. So the general notion is that local and approximate molecular clocks are more reasonable than a universal molecular clock to be applied to all living things. So that's where we're at. For a particular group of organisms, you might be able to make such an assertion that, seals and sea lions diverged at this point in time. Maybe. Maybe there was a bottleneck that affected one and not the other, and your clock measurement is skewed. It's off. So it's a controversial notion. Okay, we're going to talk about rooting. I'm taking longer than I hoped. All right, in an unrooted tree, the direction of evolution is unknown, And sort of the relationship among the distant sequences is also sort of unknown. The root is a hypothetical idea. It represents an organism which is equally distant from everything else on the tree, right? So a bacteria is about the same distance from a human as it is from a mouse, right? It doesn't work as a root for a tree, though, because you don't have orthologous sequences that you can use. You could use maybe a reptile as a root for a tree between human and mouse and dog, because there are orthologous sequences, and yet the divergence between the root or tree, the root node or organism, and all the other things you're trying to organize is equivalent. So it gives your tree an orientation. It shows you where the origin of the branch points are, and it also organizes things a little bit. So the root can be either placed on a branch or a node. So this is an unrooted tree. Oops. And this is a rooted tree, where rat somehow makes a root compared to human, mouse, and chimpanzee. Okay, so the outgroup should be a sequence known to be less closely related to the rest of the sequences than they all are to each other. It should ideally be close as possible while still maintaining that distance. In other words, bacteria won't make a good root for a tree of mammals. Something that's not quite a mammal would be nice. You know, maybe an echinoderm or a kangaroo or something like that. It has a branch point that comes in earlier than all the other branches that you're trying to resolve. The root exists actually not at that outgroup, but equidistant between that outgroup and everything else, right? It falls somewhere on that axis. So the root's over here somewhere. Okay, I wanted to add one little piece here. When you'd make a tree using, say, neighbor joining or minimum evolution, your software has to produce not just a graph, but also a data file that could be used by other software to view and analyze, redraw your tree. And so the authors of a variety of software programs came together, and they came up with a standard format that just uses a set of nested parentheses. And this tree with these distances is represented by this little string here. You know, so A and B are most close to each other, and they have the following distances from the root, which is F. C and D also form a subgroup, and then E is outside of the CD subgroup, but not part of the AB subgroup. You're not supposed to be able to read this, but some software could read this. And these Newick things can get pretty long when you have a lot of data. And they can include the distances or not. That the format is flexible about that. I think it's pretty cool. These guys worked out this format over one night of dinner at the Newick Lobster House in Dover, New Hampshire. So it's Fesselstein, Madison, Swafford. These are the authors of the major software packages that we'll talk about. And for a very long time, like 2 generations, there was just like half a dozen scientists that controlled pretty much all of phylogenetics. I mean, it's gotten a little more democratic lately, but still, it's a pretty narrow area when you talk about developing a new algorithm and selling it to the world. Okay, so if you have a data set, you've made your alignments, you've chosen a piece of software, you've either built a distance matrix and built a neighbor joining tree, or you've done a minimal evolution tree, and you've produced a tree. The tree is actually this set of brackets. The graphical representation is up to you. There's a bunch of software that can take this set of bracket numbers and draw the tree either as this sort of rectangular thing, or the branches can be made swoopy, or they can be made into V's, or they could be made into this very pretty circle. All of these trees are equivalent. They contain the exact same information. So if you think that one tree contradicts another, you're wrong. If you think that one tree visually represents the point you're trying to make better, then use that tree. Right, so here I was trying to look at the subfamilies of cytochrome genes, I think. No, subfamilies of ABC genes, ABC transporters. And the subfamilies were known, and this swoopy, round version of the tree was a lot easier to fit onto a page and allowed me to illustrate the subgroups much more neatly. So the same tree could be done as a rectangular tree, and these are outputs from Mega software. There's another program called Phylodendron that reads in these Newick formatted files and gives you 20, 50 different options for ways to print out your tree. So you can format the tree as you want and in the way that best makes your point. It doesn't change the underlying branch pattern. Okay. Is there a correct tree? Is there a right way to display my data? Is there a right way to calculate my data? It's pretty easy, actually, to use this software once you get the hang of it. Provided the data are clean, you've chosen a good outgroup, you've chosen an appropriate algorithm, you haven't too blatantly violated the assumptions, can you find a true, correct tree and prove it? No. I mean, you could do a simulation where you know the genetic distances because you took a original sequence and you mutated it into a whole bunch of subsequences, and you know how many mutations each one has. And you could test various software in its ability to recreate the tree of known sequences. But that doesn't mean that the tree you calculated from real biological data is valid, because the mutation path that those sequences followed is not the one that your simulator developed. It didn't happen monotonically. There were bursts of evolution. There was selection here and less selection there. You can't know whether the tree is truly accurate and represents the actual path of evolution. So taxonomy itself is constantly under revision. People are constantly either gathering new data or developing new algorithms and proposing new trees for the existing set of living things. Is there any way to estimate how correct your tree is? Yes. There's a thing called a bootstrap. A bootstrap, which is a general statistical technique, not limited to phylogenetics or molecular biology by any means, it randomly resamples your data. In the sense of a phylogeny, you would randomly resample the multiple alignment, leaving out some subset of the characters, right? Some subset of the informative positions where there are differences. You take, say, 90% of them and build a tree, and then randomly subsample another time with a different set of 90% of your informative positions. And do that, say, 100 times and build 100 different trees, each one of them representing the data but leaving out just a little bit. Those branches of the tree that are present in most of the bootstrapped trees, those are highly supported by bootstrapping. In other words, they're present no matter how you mung around the data. They're very stable. You know, human and mouse are just not going to be on the same branches. You know, dog and cat tend to stay together no matter how you subsample the data. Other parts of the tree come and go. It's represented in some of the trees, this particular branch point, and not in others. So those are poorly supported by bootstrapping. What's a good cutoff? Oh, people like 60%, 80% sometimes, 50% if your tree is bad. What most of the mapping software, the ones that can make this, will do is they will collapse the tree at a given percentage of bootstrap identity. So you've done 100 bootstraps, you've recalculated your tree 100 times. Any branch that's not present in at least 50% of your trees is eliminated and made flat, right? So there's now no distinction between A, B, and C. We don't know whether A is closer to B or closer to C. They all three come back to a single joining point. So that collapsing happens at every place in your tree that's not highly supported by the bootstraps. So what you get is what's called a bootstrap consensus tree. So you can just, and you can do that. You can collapse it and make a consensus, or you could show the full tree and you could put the bootstrap values in at every branch point to show how well supported that branch is. And that's a better way of doing phylogeny, because at least it shows you how robust your conclusion is with respect to the data. All right, one more thing I'm going to talk about that's related to this is this idea of selection and synonymous versus non-synonymous substitutions. And this is essentially another layer of information that's present in your multiple alignment. For each DNA substitution in a coding region, obviously, we can observe if it changes the corresponding amino acid or not, right? There's the 3-letter codons. There's an average of about 3 different codons for each protein, each amino acid, but some amino acids have fewer and some have more. A given DNA change may or may not change the amino acid of its codon. That is a synonymous, meaning doesn't change, or non-synonymous change. What we're interested in terms of selection is whether the number of synonymous changes versus non-synonymous changes differs from a random background. If the DNA changes happen at random and selection did not occur, right, then the number of synonymous versus non-synonymous changes is the same as what you would see if you just mutated the DNA randomly. If the particular sequence is undergoing conservative selection, meaning the protein has a shape and many of the amino acid changes are deleterious and therefore eliminated, the organism that carries that mutated protein has lower fitness, et cetera, then you will see an increase in synonymous changes versus non-synonymous changes. And that's very common. I showed you that map in Cladistic, where you see the relative conservation of the bases in a given position. You could see a very similar map of synonymous versus non-synonymous, where the most conserved parts of the protein have a higher level of synonymous versus non-synonymous changes. This thing is called, so the synonymous are Ks, the non-synonymous are Ka, and there's a little formula for this that computes the ratio of synonymous to non-synonymous as compared to random. It's always as compared to a random background. What's very interesting is occasionally you see a region where the number of non-synonymous changes is elevated. The protein is changing more rapidly than you would expect just by chance. Mutations that cause amino acid differences are being positively selected. When would that happen? Well, let's say it was a surface protein of a virus and it's evolving to evade immune detection. Or it's an immunoglobulin and it's evolving to randomize the number of possible antigens that it can recognize. A few other situations where positive selection is observed. There's a more controversial scenario where a particular protein is under heavy selection pressure to adapt to a particular solution. A bacteria is under attack by an antibiotic. The antibiotic recognizes a particular enzyme. That enzyme needs to mutate in order to escape antibiotic pressure. that might be a positive selection for changed amino acids in that the gene that codes for the target of the antibiotic. That's a scenario that makes sense. So the Ka-Ks ratio tests for the signature of selection. And you could pass that over any codon-based alignment, right? It's better to align first the amino acids and then substitute in the DNA sequences, because if you align just the DNA, you could easily shift away from the proper codons. And Ka-Ks varies within a gene. So this would be an active site where selection is more prevalent, and over here it's closer to 0, where there's no selection. All right, very briefly, the ancestor of all the phylogeny software is Phylip. It's very powerful. It's free and open source. It is command line software. It has many subunits, and it is devilishly hard to use. If you have a good command of Phylip, you are employable as a molecular evolutionist anywhere. Clustal is very easy to use software, and it can make a neighbor joining tree, and that's all the phylogeny it can do. Mega is free, Windows favoring, but does function sort of on Macs, software that incorporates all of the methods that I've described today. It does neighbor joining, minimum evolution, and maximum likelihood. It draws trees. It has built-in Clustal. Very handy, useful tool. If you can do Mega, you can be the evolutionary tree builder for your lab. You may not, make it on the national stage, but you'll be able to build good, also supportable trees. In other words, with Mega, there's a little button that prints a log of exactly what you did to produce the given tree with a given sequence alignment. So it tracks all your steps, and you could paste that into the into your figure caption, and you're done. And it also has another button for citation. Boom, paste that into your citation section, and you're good. Mesquite was a previous attempt at a do-everything phylogenetic software. I found it really hard to use. POP is a commercial software that implements parsimony primarily. That's what POP stands for, is phylogenetic analysis using parsimony. It's commercial, it's cheap, relatively hard to use, not quite so hard as Phylip. But again, very strongly supported by the reviewing community. So if you've done your trees with POP and you document what you've done, you're probably going to be positively reviewed. And Philodendron is a tree drawing tool that can make pretty pictures once you've built a tree with something else. Other web resources you can look up yourself. All right, so given all these methods, how can you be reasonably sure that you're doing a good job? What method should you use to analyze your data? I would say look at papers that are published in your field. What methods have they used? If you're working on viruses, there's probably a different set of software than if you're working on mammals or human anthropology or whatever it is that you're working on. So, see what people are doing. In some cases, different methods can be used synergistically. Neighbor joining is quick, and it generates a halfway decent tree. That tree can be used as a basis for minimum evolution or maximum likelihood methods. To narrow down the universe of all possible trees to be tested, start with a tree that's already been built with a reasonable method on the same data. If you use several different methods on the same data and you get similar trees, that's good evidence that your tree is valid, and you could submit such a thing. Some software even allows you to find the consensus between two different trees. Okay. Oh, wrong summary. My bad. Okay. Analysis of variance comes next.